Hi, my name is Jim Valcourt, and I'm a drug discovery fellow at D.E. Shaw Research here in New York City. And today, I'm excited to tell you about some of the work that I did during my PhD in the lab of Sharad Ramanathan at Harvard University. The human body is made of around 40 trillion cells, and all of those cells are specialized so that the cells in your brain, for example, are quite different from the cells in your skin or those that line the gut. Now, all of those cell types, despite their differences, come from a single population in the early embryo, known as the pluripotent stem cells. Those pluripotent stem cells, shown in white on this slide, have to make a series of decisions by listening to signals from their environment. So they might first choose between being a blue cell or a yellow cell, and those blue and yellow cells will then go on to make a series of further decisions, again listening to signals from their environment, to produce that diversity of cell types seen in the adult body. Once a cell makes a decision, it actually loses the ability to adopt alternative fates, to change its mind. So once a cell becomes one of these blue cell types, it actually loses the ability to become a yellow cell type. So we would call that cell committed to the blue lineage. Now, I study this process of commitment in the context of one specific cellular decision. In fact, the first decision that pluripotent stem cells have to make, either between an ectoderm cell, shown here in blue, which is the precursor to things like skin cells and brain cells, or mesendoderm, shown in yellow, which is the precursor of muscle cells and gut cells. Developmental biologists frequently think of this process of a cell choosing between fates as being something like a ball rolling down a hill. And we call this hill the Waddington landscape, named after the first scientist who drew this cartoon back in the 1950s. So the pluripotent stem cell is represented by this ball, and it starts up at the top of the hill, and it's choosing between one of these two different valleys, which corresponds to different cell fates. And in this picture, giving that cell a signal that it should become for example, a yellow mesendoderm cell, corresponds to giving it a kick to the right on this landscape. So let's follow one of these cells as it makes a decision. So our first cell might start moving down that ectoderm trajectory because it's getting some signals from its environment that makes it want to turn into a blue cell. But if we give it a yellow signal, a kick towards the right on this Waddington landscape, it's still able to roll over into that mesendoderm yellow valley because there's no barrier preventing it from doing so. However, if we look at a different cell, one that has moved a little further down that ectoderm blue valley before it gets the kick towards mesendoderm, it instead runs into that barrier between the two fates, between the two valleys. And so that cell, even though it gets the same kick towards mesendoderm, is no longer able to overcome the barrier between the fates, and it has committed to that blue ectoderm lineage. So our goal was to measure and control when cells commit to a particular fate, in this case to that blue ectoderm fate. To that end, we drew inspiration from the Waddington landscape, and we measured two things. The first is how far along that blue ectoderm path a given cell is. And the second thing we measured was the cell's ability at each point along that path to transition over to the other alternative mesendoderm fate, if given the proper signal. In order to make this measurement, we had to genetically engineer our stem cells to glow different colors, to fluoresce under an ordinary light microscope. So we used genetic engineering to tag two proteins that are very important in this process. The end result is that our stem cells show up as white on the microscopy images that I'm about to show you. As they transition down the path towards the blue ectoderm fates, they will actually glow blue. And then, if they're able to transition over to the yellow mesendoderm fates, they will glow yellow. This video shows a population of stem cells. Each of those little dots is the nucleus of one of those cells. And as they grow and divide, I'm giving them a signal that tells them to become blue towards that ectoderm fate. And so you can see they're becoming more and more blue as time goes on. And then right here, I give them a signal that tells them become yellow. Some of the cells are able to respond and turn into that yellow mesendoderm fate, but some are committed to that blue ectoderm fate. So now we can follow individual cells through that video I just showed you. So on the y-axis, I'm going to show you the color of the cell over time on the x-axis. And we're going to ask whether we could have predicted whether the cell would be able to respond to that signal by becoming a yellow mesendoderm cell or not. And so let's follow the first cell. It starts off as a white pluripotent stem cell. Through the first half of the video, it becomes more and more blue in response to its environment. And then at the white dotted line, I give it that signal that tells it to become yellow. And this cell is able to do that. It turns around, changes its mind, and turns into a yellow mesendoderm cell. In contrast, this other cell from that video, again starts off as a white pluripotent stem cell, becomes more and more blue through the first half of that video. But then at the white dotted line, I give it that same signal, and it's not able to turn around and become yellow. It stays blue. And so if I do this for 100 different cells from that time lapse, you can see a pattern start to emerge if I color each of the trajectories with the final fate of the cell. So all of the yellow lines are the cells that ended up turning into yellow mesendoderm, and the blue lines are the ones that finally ended up becoming blue ectoderm. And you can see that at the white dotted line, at the point of signal addition, the cells that end up yellow were already a little more yellow than their neighbors that ended up blue. And that allows us to actually physically sort out populations that are able to become yellow from those that have already committed to the blue ectoderm lineage and ask what makes them different from one another. So by comparing the committed cells to the non-committed cells, we were able to uncover a core gene regulatory network that governs whether or not a cell has committed to its fate or not. And by perturbing these genes, we found that we could control the cell's fate in one of two distinct ways. The first way is controlling sort of how far along that ectodermal valley the cell moves. And the second way was we could actually change the barrier between the ectodermal and mesendodermal valley entirely. We could control the shape of the Waddington landscape itself. So we found that there are in fact these two knobs that evolution can use to control cell fate. We think these findings are important for a couple different reasons. First, this mechanism may be important for setting the size of different tissues in the developing embryo, which is crucial for proper development. Second, knowing the existence of these two knobs may allow scientists to disentangle the mechanisms behind cell fate control during development and allow them to make specific cell types to treat disease with stem cell therapy. And finally, we think that ultimately understanding the mechanisms of how these processes are regulated during normal development may one day help with therapeutic applications for conditions where these processes are dysregulated, such as in developmental disease and in cancer. Finally, I want to take the opportunity to say thank you to everyone who made this work possible, especially my wonderful collaborators and colleagues. I'd also like to extend a deep thank you to the staff of Harvard Horizons, who all worked tirelessly to make this event possible. Thanks very much for listening.